In the thylakoid membrane, light hits photosystems 1 and 2 simultaneously. In photosystem 2, the light excites electrons, and all the excited electron energy will be transferred to the reaction center chlorophyll. The reaction center chlorophyll will pass it on to proteins in the thylakoid membrane. Electrons lost by photosystem 2 are replaced by photolysis. This takes electrons from H2O and makes oxygen. As the electrons are passed through the electron transport chain, energy from the electrons are used to pump hydrogen ions from the stroma to the thylakoid. This makes a concentration gradient. This powers ATP synthase that turns ADP into ATP. Low-energy electrons from photosystem 2 are moved to photosystem 1. In photosystem 1, electrons are reorganized and passed through the electron transport chain to make NAD+ into NADPH. Carbon from CO2 binds with RuBPH by an enzyme called Rubisco to make a 6-carbon sequence. The 6-carbon chain splits into 2 3-carbon chains. That is called PGA. Then, ATP provides energy and NADPH adds 1 hydrogen ion to each chain, making them G3P. 12 G3P are made at a time, meaning 36 carbons exist for RuBP. 2 of the 12 G3P are given off to make glucose. The last 10 G3P are given to make RuBP. 3 of the 10 G3P are put together, making a 9-carbon chain. 5 carbons are given off to make RuBP. Then another G3P is added to the remaining 4 carbons, making it a 7-carbon chain, plus another G3P to make more RuBP. In the energy investment stage, 2 ATP give energy to the glucose molecule. This makes a 6-carbon sugar diphosphate molecule. The molecule splits and the 2 3-carbon molecules are converted to pyruvate. ATP is formed from this. 2 ATP, 2 pyruvate, and 2 NADH are produced. When O2 is present, pyruvate and NADH enter the mitochondria. Each pyruvate is converted to a compound called acetyl-CoA. During pyruvate oxidation, electrons move to NAD, making NADH. A carbon is lost, making CO2. Then, acetyl-CoA will bind with a 4-carbon molecule, making it a 6-carbon molecule. Then, NAD+ comes to it and makes it a 5-carbon molecule, letting go of CO2 and making NAD+ turn into NADH. Another NAD+ comes to it and makes it a 4-carbon molecule, letting go of one CO2 and making NAD+ turn into NADH. Then ADP combines with another phosphate, making ATP. Then FAD combines with it, making FADH2. Then NAD+ combines with it, making NADH. For every glucose molecule in glycolysis, the cycle completes twice. Oxidation in the Krebs cycle yield 8 NADPH, 2 FADH2, 2 ATP, and 6 CO2 for each glucose. Electrons are transferred between the membrane proteins. The cell is able to capture energy from it, and this produces ATP. The membrane carriers pump hydrogen ions across the membrane through ATP synthase, which synthesizes ATP. Oxygen acts as the terminal electron acceptor. When it accepts electrons, it forms water. All the carriers from earlier bring their electrons into the chain. From this, most ATP from cellular respiration is made. Altogether, the electron transport chain makes 32 to 36 ATP per glucose molecule.